Hi, this is another little clip on transport. Here we have 4 different diagrams, and you can see in diagram A, you have particles moving from a higher concentration to lower concentration in diagram A. It's moving through the phospholipid bilayer and there's no energy required. Diagram A is showing you simple diffusion. Diagram B is showing you facilitated diffusion because you don't need energy to move those particles, again, from high concentration to low concentration. But what is different is you have this protein channel or an integral protein that facilitates or helps to move some of those materials through. These are both types of passive transport, again, because they're moving from high concentration to low concentration. It's also said that they move with the concentration gradient. They move down that slope. Diagram C is showing you simple diffusion of water, which we call osmosis. Now, sometimes osmosis requires protein channels, but it is passive transport. So when you talk about hypertonic, hypotonic, and isotonic solutions, it's always dealing with osmosis, and osmosis is a type of passive transport. In diagram D, as a review, particles are moving from low concentration to higher concentration. In order to do that, ATP, adenosine triphosphate, or energy is required. If you recall the car on the slope, if you have a slope on the low end of the slope, you have to turn that car on or push that car up the slope or up the concentration gradient. That requires energy. If we go to the next activity, we have exocytosis and endocytosis. Now, this is a type of active transport. This is when the cell uses vesicles to move larger materials into and out of the cell. And vesicles are typically created by the Golgi apparatus, or they can be created by the cell membrane. The cell membrane, in order to take larger materials in, will engulf the larger materials, and that circular structure is your vesicle. Okay, endo and exocytosis. One way to remember which you have EX, exocytosis, for exiting the cell, and then you have endocytosis, meaning it enters the cell through vesicles. So what is the vesicle? These vesicles are, let me just label that for you. Your vesicles are right here, and you're going to see those in the diagram below. There's a vesicle, there's a vesicle, and I like to think of vesicles as cellular vehicles. So vesicle, vehicle, that's how I remember it. If we go down to the bottom for this diagram, it's showing exocytosis, and you can see the vesicles transporting materials outside of the cell. The Golgi apparatus makes vesicles for various reasons. One reason the Golgi apparatus would make a vesicle is when proteins are folded and made, and they're sent to the Golgi apparatus. The Golgi apparatus is going to package that protein in such a way so that the cell can use it. If the cell can't use it, it is sent to the lysosomes, and the lysosomes break it down and send it outside of the cell through that secretory vesicle. And again, this is showing you exocytosis. It's a type of active transport. It does require ATP, or adenosine triphosphate, and I hope that is helpful.